Welcome, Miss Hannah.
Okay, so hello. Good afternoon, everyone. Thank you, everyone, for taking the time and effort to be here. Actually, this is the first time that I get to speak with scientists coming from diverse fields and expertise. So I really would like to take this chance to learn from all of you. So at the end of the talk, I really would love to hear your thoughts and questions or ideas that you may have. So today we will talk about a biological phenomenon called large-scale cell death that is frequently found to occur during development. It functions to eliminate millions of cells in order to shape the tissue structures and organs to their appropriate forms. Classical examples include the interdigit regression and also cell death that shapes the tubular structure for some of our organs, and also the regression of the tadpole tail. Now, all of these death events observed in the 19th century were mostly attributed to a type of cell death called apoptosis, first coined by Robert Horvitz. Here they used C. elegans as a model system and found that apoptosis functions to trim lineage-specific cells that are no longer needed for the mature organisms. However, in higher organisms like vertebrates, there seems to be a different phenomenon in that cell death happens in large scales, that it could even eliminate organs and tissues involving millions of cells. This was first observed by John Saunders in the 1960s, that is drastically different from that observed in C. elegans, which brings us to this question, century-old and few have pursued: how does large-scale cell death occur? Or in other words, how is cell death coordinated in space in order to eliminate vast populations of cells? So here I hypothesize that large-scale cell death could be mediated by propagating waves of cell death signal. And this idea resonates with this observation made by John Saunders, wherein he described waves of cell death happening in the developing limb. So with this in mind, I searched for the type of cell death that could have propagating activity in space. So here I am showing you a monolayer of human epithelial cells that are under metabolic stress. Specifically, I deprived the cells of the nutrient cystine. Here, each dot is a cell nucleus that would light up upon cell death. So please focus your attention to the right-hand side, where you would see cell deaths initiate and propagate across the whole cell population. And with this initial observation, I developed an assay to be able to quantitatively study this phenomenon. Here, I could use light elimination or photoinduction to control the initiation or what timing and spatial location it would initiate. So here, I did photoinduction at this local region, and you would see cell deaths to initiate and propagate across these thousands of cells at a constant speed, as indicated by this constant progression of the cell death boundaries. Now here I want to emphasize that even though people previously found that cell deaths could be transmitted from one cell to its neighboring cell, it is the first time that we could observe cell deaths propagating across thousands of cells without any signs of slowing down. And this type of phenomenon is what we call trigger waves. In contrast to diffusion, wherein the signal's intensity would quickly dampen in space, trigger waves are able to maintain the signal's intensity as it propagates across distance. And this allows for the propagation of activities at constant speed over large distances. And this very nature of a trigger wave allows for cell deaths to propagate in a spatially infinite manner. So today I want to talk about how nature could utilize this propagating cell death for sculpturing our tissues during development. And furthermore, we venture into the possibility of harnessing this propagating cell death for cancer therapy. But before we go into this, we first want to know the basic ingredients that allow for cell death trigger waves to occur. And with this, I want to make an analogy to wildfires. So just like wildfires, trigger waves are composed of these three components: an initiation point, a spatial coupling mechanism, or a mechanism by which a signal could be transmitted from one cell to the other. And third is the key component for trigger waves, which is the positive feedback loops. So this is analogous to the fuel for the fires. So it can be the trees and grasses that regenerate the fire as it travels. So in a cell population, it is the positive feedback loops that act to amplify this signal as it diffuses from one cell to the other. So you can imagine that this signal will not get any lower in its intensity because every step of the way, it is regenerated by this positive feedback loop. Now, due to the interest of time, I could only share with you about this positive feedback loop component and what we think could be the signaling molecule that could spread this cell death. So I think one useful information is that this propagating cell death is unique to a type of cell death called ferroptosis, mediated by iron and reactive oxygen species, or ROS, accumulation. So to induce ferroptosis, we are depriving the cells of the nutrient cysteine, and this cysteine is necessary for synthesis of glutathione. And depletion of glutathione leads to accumulation of lipid peroxides in the cell membrane, which leads to membrane rupture and eventually ferroptotic cell death. So we hypothesized that this lipid peroxidation could be one of the signaling molecules that would spread between cells and cause cell death. So in this experiment, I monitored the lipid peroxidation in real time. So again, I can do photoinduction at this local region, and this photoinduction caused cell death propagation. And you will see this wavefront of lipid peroxidation shown in the yellow color that proceeds before cell death shown in blue. And this lipid peroxidation, here LOOH, is one of the multiple reactive oxygen species that I found to be elevated at the wavefront. And these ROS species interact with each other through multiple positive feedback loops mediated by NADPH oxidase and iron. So I can test this model by perturbing the different components of this feedback system and see how this would affect wave propagation. I can only show you one example where I will modulate iron levels in the cell population. So in this example, again, I can induce cell death propagation. And after some time, I would add iron chelator. And you would see obvious slowing down in the wave speed. And this is also apparent when we look into the progression of the cell death boundaries. And on the other hand, I can also supplement iron. So after photoinduction and wave propagation, I would then supplement iron. And you will obviously the increase in the wave speed. And likewise, I can also, in real time, yes. So I can also perturb the other ROS feedback loops and see how this would, and I did see an alteration in the wave propagation speed. So with these results, we have now shown that cell death could propagate as trigger waves of reactive oxygen species that are amplified in space. So this nature of trigger waves to propagate cell death without spatial limits sounds really terrifying, right? I mean, like once cell death initiate, then we will all end up like skeletons, which means that this trigger wave of cell death must be regulated. Inspired by John Saunders' observation, I looked into the role of ferroptosis during limb development, using the chicken embryo as a model system. And what I found is that lipid peroxidation, a hallmark of ferroptosis, is actually widespread in the developing limb. So here, lipid peroxidation shown in green is actually very abundant at this central region of the limb. And at the transverse section, you will see lipid peroxidation happening out at the outer layer and extends to the inner layers and overlaps with the muscle cells. And we also note the presence of lipid peroxidation at this interdigit region. So this shows that ferroptosis indeed happens in large scales during limb development. But does it happen as a wave? So here I developed an assay to monitor cell death in real time in the ex vivo developing limb. So as you can see here, cell death indeed propagates across the developing limb in this central region here, which is also where we found lipid peroxidation in the previous slide. And this cell death is ferroptosis because when we applied ferroptosis-specific inhibitor, UAMC, we see a significant decrease in the cell death area. And what's remarkable here is that even though cell death propagates and is quite massive, it is spatially restricted to the central regions only. And the cells at the lateral region, they remain viable. So how is this possible given the nature of a trigger wave? So one explanation could be that the fuel for the fire or the fuel for cell deaths, which are oxidizable lipids, could be spatially patterned. So here I measured the amounts of oxidizable lipids, and indeed what I saw is that there is higher oxidizable lipids at the central region where cell death waves occur. So this means that oxidizable lipids are spatially patterned so as to restrict cell death to eliminate certain populations only. And furthermore, I assayed for the functional significance of ferroptosis during limb development by in ovo injection of ferroptosis inhibitor. And one striking phenotype I observed is that there is a compromise in interdigit regression as compared to the untreated control limb. The ferroptosis inhibitor-treated embryos show this tightly joined digits. And another phenotype I observed is an alteration of the muscle remodeling process. So whereas in control, the muscle bellies are well aligned with each other and are well segregated from each other and extending to each digits. However, when ferroptosis is inhibited, this segregation is compromised. And you could see much higher numbers of muscle fibers. To the right is a quantification of the muscle fiber counts from different embryos where you can indeed see much higher muscle fiber counts when ferroptosis is inhibited. So altogether, these results show that the propagating cell deaths could indeed be utilized in order to eliminate these unwanted cell population as the embryo develops. So now we wonder if we could utilize this propagating cell death for cancer therapy. So I think one great challenge in conventional chemotherapy is the partial drug response, which would lead to drug resistance. So in our experiment, where I treated cells with different chemotherapy drugs, that after 2 days, we could only see about 40 to 70% cell death. However, when I treat the cells with ferroptosis inducer, we see this rapid rise in cell death that would eliminate 100% of the cell population. So with the goal of using ferroptosis for cancer therapy, we tested more than 20 different cell lines of 12 different tissues of origin and found that all of them could exhibit ferroptosis waves, which is very encouraging. However, there are also challenges here. So when we quantitatively analyze the wave propagation behavior in different cell lines, we found that there is a great variation in the tendency of the cell population to undergo population-wide cell death. So let me give you 2 examples. Like in kidney adenocarcinoma cells, you will observe cell deaths to eliminate the whole population. However, in bone osteosarcoma cells, we observe that cell death could only happen to a portion of cells and leaves some cells to survive. So how do we overcome this problem? Our next goal is to be able to convert these resistant cell population and partial responsive cells to undergo complete eliminations through cell death waves. We plan to apply multi-omics approach and also unbiased approaches like CRISPR screening in order to know better about the molecular networks that makes these resistant cells resistant. And by rewiring these molecular networks and also enhancing the ROS feedback loops, we might be able to induce propagating cell death waves in these resistant cells. So with that, I want to thank my advisor, Shen, for all of his support and all his mentorship that made this project possible. To Xiacho Wu, he I actually shared this work with Xiacho. He worked with me on the computational aspect and also imaging analysis. And Hao Kun-Lin and Vivian, they actually initiate the direction towards cancer therapy. And also I want to thank my lab mates for the village that really makes this project possible, and also to the scientists in IMB who helped us through some experimental technicalities. And we thank the funding source, and thank you everyone for your attention, and I'm open to receive any of your questions and suggestions.
Thank you, Ms. Hannah.